Okay, it is noon, so we are going to get started. Welcome everybody to the Sampling Research Farm Irrigation and Nutrient Management Virtual Field Day. Thank you all for attending. We would obviously all prefer to be attending in person, but that's not possible under the current circumstances. So we have 5 presentations for you today. We're going to do our best to stay on schedule. Everybody is allotted 15 minutes for presentation time, and then we'll have brief question and answer period following the presentations. If you have questions as we are going, please enter them through the Q&A feature. You have the ability as webinar participants to upvote questions or like questions that you really want to see answered. So please feel free to ask away and we will do our best to get those questions answered for you. If we run out of times for a particular presenter to answer questions, we will allow more questions at the very end of the webinar. So with that, I will turn it over to our first speaker, Vasudha Sharma, who will be presenting on irrigation and nitrogen management research projects for profitable corn production and groundwater quality protection.
Thanks, Emily. I'll share my screen. All right. Can you see my full screen?
Yes.
Okay. All right, so welcome everyone to our Sandplain Research Farm Virtual Field Day. My name is Vasudha Sharma. I'm the Extension Irrigation Specialist in Soil, Water and Climate Department at University of Minnesota. And today I'll be talking about my irrigation and nitrogen management research projects that I'm conducting at the farm this year at the Sandplain Research Farm in Becker. So talking about crop irrigation in Minnesota, we know irrigation is essential for crop production in the central sands region of Minnesota, mainly because of the low water holding capacity of the soils and at the same time, low precipitation during the critical crop growth stages. So in these soils, insufficient or low irrigation can lead to huge grain yield losses as well as lower the grain quality. However, in the recent years, what we have seen is that water quality under these sandy outwash soils is declining because of the presence of nitrate, and that has been steadily increasing under intensively farmed lands. And that's because over or excessive irrigation in these soils increase deep percolation of the nutrients and is also huge waste of energy and water resources. And at the same time, it has a negative impact on grain yield because of loss of important nutrients from the soil. So through our irrigation and nitrogen management research projects, our main goal is to develop and evaluate the strategies that reduce irrigation-induced nitrate leaching and conserve water resources, and at the same time do not significantly impact our crop yields. So right now, I'm focusing firstly on the most important aspect of irrigation water management, that is scheduling of irrigation. And then I'm also trying to understand some advanced irrigation management strategies like deficit or limited irrigation management in Minnesota. So these are the 2 research projects that right now I'm conducting. So in our first project, what we are doing is we are evaluating the impact of 4 main irrigation scheduling methods on corn yield and nitrate leaching. That will help us in understanding the pros and cons of each of these methods. So the 4 methods that we are comparing are listed here on the screen. The first 2 methods are weather-based irrigation scheduling methods, which is checkbook method and irrigation management assistant tool, IMA tool. The third is the soil moisture monitoring, where we monitor soil moisture using soil moisture sensors, and based on that we irrigate. And then fourth one is the EPIC crop growth model. This is a crop growth model that simulates irrigation based on previous data and weather data. So if we look at the soil water balance in the screen, in the figure that you see that shows all the inputs and outputs of water balance, what we really want in efficient irrigation is trying to manage this soil water in the root zone that you can see. So keeping the soil water in a range, the range is between field capacity and management allowable depletion, that it does not leach or have a lot of deep percolation, and is still adequate to supply the transpiration and evaporation, both evapotranspiration demands of the crop. So the decision we make that how much irrigation water we need to apply and when to apply is based solely on this soil water, that how much water at the end is present in my rooting depth, in my root zone. So in our soil moisture monitoring irrigation scheduling method, in our study, we are using a neutron moisture meter that measures soil moisture up to 4 feet depth. And then throughout the growing season, we measure this soil moisture from our soil moisture monitoring plots, and then calculate the soil water depletion in the root zone. And based on that, we irrigate. So that soil water that I showed you in the previous slide, that soil water is being measured using soil moisture sensor in this method. In our weather-based methods, they use daily water balance equation, both checkbook and irrigation management assistant tool. They use this daily water balance equation that you can see on the screen. So the current day deficit, that is denoted by DC, is equal to the deficit that happened on the previous day, DP, plus the crop evapotranspiration, the crop water use, ETC, minus precipitation, minus irrigation. So it takes into account all the inputs of water and all the outputs of water, and then estimate this soil water in the rooting zone. So in both of these methods, the soil water is not measured as we measure in soil moisture plots. However, it is estimated. So both of these methods work on the same principle. There are some differences in these two methods in the ways it calculate the evapotranspiration, that is crop water use. Another major difference is that we need to manually log the data in our checkbook method, whereas in IMA tool, we do not need to manually do anything at the beginning of the season. We just give it all the inputs, and then it recommends irrigation over time. So in addition to our soil moisture measurements, we also collect plant samples and soil samples from all our plots throughout the growing season to understand the nitrogen dynamics. And we also measure nitrate leaching below the root zone at 4 feet from all our plots to understand the impact of irrigation methods on nitrate leaching. So there are some results from 2019. This is the second year of our study. Last year was the first year. In 2019, which was the very wet year, we received around 25 inches of rain in the corn growing season. The bar graph on the left here indicates the irrigation amounts in each treatment. So we irrigated maximum in soil moisture monitoring, which is about 4.9 inches of irrigation, followed by checkbook method, where we irrigated 4.2 inches, and then EPIC crop growth model, where we irrigated 2.8 inches. The IMA tool recommended only 0.8 inches in 2019. The graph on the right shows you the seasonal water balance evapotranspiration from each treatment. What we see is that there is no significant difference in evapotranspiration between treatments. However, it followed the similar trend as irrigation. So if you look at the trend of irrigation amounts and seasonal ET, it follows the same trend. The high irrigation treatments has high ET and low irrigation treatments has low ET. So what we can really tell from this data is that even though there was a big difference in irrigation amounts between treatments, the difference in ET was not significant, which means that there was enough water in the root zone and there was more water mining in the low irrigation treatments like IMA and EPIC as compared to high irrigation treatments. And this is also indicated by the grain yield results. We did not find any significant difference in grain yield between soil moisture monitoring, checkbook, and EPIC crop growth model. However, IMA yield was significantly lower than the EPIC treatment, which shows that lower irrigation amounts in IMA reduce the yield, but that does not mean that higher irrigation amounts increase the yield, because the highest yield, if you see, was obtained in the EPIC crop growth model, which is not the highest irrigated plot. Irrigation was highest in soil moisture monitoring. So there is something else other than water that is playing a role, and we think that could be nitrogen. So the maximum nitrate leaching was observed in the checkbook method. So the 4 lines are 4 irrigation scheduling methods. The checkbook method is indicated by these yellow triangles on the top. The total irrigation amount was slightly greater in soil moisture method than in checkbook method, still higher nitrate leaching. It might be due to the timing of irrigation. So if you carefully look at in this circle, the main difference in nitrate leaching between the checkbook and soil moisture monitoring started to occur on around July 30th. It's the time when the checkbook method recommended high irrigation, but no irrigation was recommended by soil moisture monitoring method because there was enough water in the profile according to soil moisture sensors. This table shows you the nitrogen uptake by plants in each treatment at the end of the growing season. And again, we found that the checkbook method has the least amount of nitrogen uptake, which could be due to high nitrate leaching. So the take-home message from all this data that we collected in 2019 is that even in the wet year, like last year, irrigation amount and timing both can have a significant impact on nitrate leaching, nitrogen uptake, as well as on crop yields. But this is only 1 year of data. We have collected data this year as well, and we'll present that once we have all the data for this year. So in the same project, we are also comparing the performance of different soil moisture sensors. I'm not going to talk about this in detail today, but one thing that I want to focus is this new sensor node that Bryan's team developed, and they're going to talk next about it. So we have installed 16 units, 16 of these this year in our plots. This is a new initiative that, as I mentioned, Bryan is going to talk today. So moving on, the second research project that I'm focusing on is to understand the effect of irrigation rates and nitrogen rates and their interaction on corn yield and water quality. The specific goals of this research are to evaluate variable rate irrigation and nitrogen rate interaction effects on various crop parameters. And the second main objective of this research is to explore the remote sensing and proximal sensing data to detect water and nitrogen stress in plants. So we are studying the interaction of 4 irrigation levels and 6 nitrogen rates. There are 24 irrigation nitrogen combination treatments that are replicated 4 times. So there are total 96 plots in this study. This is the first year of the study. We are going to continue it for 2021 and 2022. So our research here follows a split plot experimental design. The red boxes here indicates the replications, and within each replication, we have 4 main irrigation plots indicated by these white boxes. And then within each irrigation block, we have main plot, we have 6 nitrogen rates. The main thing that we did this year was installing 96 lysimeters permanently in this study. So by installing permanently means that we do not have to take them out for harvesting or other field activities. This permanent installation, ensures that our data is accurate because there will be very less soil disturbance every year, as well as with 96 lysimeters, we have great spatial resolution that we can accurately quantify the nitrogen loss. So on the screen, you can see some pictures where in the first picture, we are digging these long trenches in our plots, and then digging these holes, putting the lysimeters in, packing the soil. And then on the fourth picture, you see we have connected all the lysimeters together through this PVC. And then in the fifth picture, these are the outlets. So from each outlet, we can sample 6 lysimeters, and we do this sampling every week and collect the water sample and send it to lab for nitrate nitrogen analysis. Again, for each treatment, we are measuring soil moisture. So from 72 plots in this study, we are measuring soil moisture every week to understand the water dynamics. In addition to soil moisture and nitrate leaching measurements, we also take multispectral images throughout the growing season using unmanned aerial vehicle and proximal sensor data. Through this data, our idea is to correlate this remote and proximal sensor data to soil and plant monitoring data to develop some correlations that can be used for variable rate irrigation and nitrogen management in the future. As I said, this is the first year of the study. I do not have data to show you, but in summary, what we expect at the end of this research is to have some data and information that helps us in understanding how corn respond to different irrigation and nitrogen fertilizer rates and their interaction, and which will help us in developing the production functions. And also this research provides us data and information on fundamental relationships that helps us in advancing the sensor-based research, both on irrigation and nitrogen. At the end, I would like to acknowledge all the funding agencies who are funding these projects and our field crews, both at Sandplain Research Farm, Ron's team, and our soil, water, climate field crew for assisting us with field work. Thank you so much. I can take any questions you have now. You can also contact me through my email, and you can go to our extension website where we have good stuff about irrigation.
We have a question from John who says, does your 50% irrigation mean 50% of readily available water? In other words, a deficit irrigation strategy?
So our 50% irrigation, so our 100%, I'll start with 100%. 100% irrigation is bringing our soil moisture back to field, close to field capacity whenever our soil moisture is approaching MAD, which is management allowable depletion, and we change this MAD value throughout the growing season. 50% is just cutting the amount of irrigation to 50% than what we were applying in 100% irrigation. So if we are applying 1 inch of water in 100%, we are applying 5/10 of irrigation in 50% irrigation.
Okay, it doesn't look like any other questions are rolling in. So with that, we will turn it over to Bryan Runck, who is a geocomputing scientist at the University of Minnesota. And he's going to present on GEM Informatics, soil to satellite, and sensor to supercomputer.
Well, thank you, Emily. Say, Emily, it's not letting me share my screen. Would you be able to allow that?
Yep.
Thank you. Okay. All right. Well, thanks so much, to Vasu and Ron and Emily for having me here today. It's just a pleasure to speak at extension events. I'm a farm kid from Southwest Minnesota, not irrigated ag there, but the experiment station was always very close at mind and a big part of our lives there in Lamberton. So my goal today is to give you kind of a broad look at what's called the GEMS Informatics Center. And then also to illustrate in a very specific way, kind of the sensing and actuation work that I lead within GEMS. I'll go into all the details of that in just a bit, but there's going to be 4 kind of big parts to this talk. I'll start off by giving kind of this high level overview of GEMS in our programs. Then I'll go in and describe specifically about the sensing work that the program has started with. And then I'll kind of take you through a story of how we went from, really a quick prototype of our initial systems and then into where we're at now. So to start off with, what is GEMS? GEMS is a joint initiative between the Minnesota Supercomputing Institute and our College of Food and Agriculture. And so, and really, we're a security and privacy first data collection, sharing, and analytics ecosystem for agriculture. And so we're very research tuned, but we also collaborate extensively across large Fortune 500 companies such as PepsiCo, and as well as small startups, and research scientists at many different universities. When we say that we're security and privacy first, what do I mean by that? On the technical side, what it means is that all the data that we have is actually stored in the same physical location as all of the University of Minnesota health record data. And so you get the same kind of physical security around that computing infrastructure. And then all of the employees at GEMS are trained in HIPAA data standards and HIPAA data management standards. And so we bring that same sensibility to any of the farm data that we interact with. On the legal side, which is really important as a public institution, because we are subject to Freedom of Information Acts. And GEMS leadership really began working probably 3 or 4 years ago with the state legislature to pass a bill that makes all identifiable farm data private, and that's on a University of Minnesota server. So we have this legal protection in place, and then we also have the state-of-the-art technical protections. The GEMS Informatics Center, so GEMS stands for Genetics by Environment, by Management, by Socioeconomics. And so this is kind of heuristic as to how we can think about agricultural systems. And really we have 4 main areas of work that we engage in. One is our core platform, which is really targeted at data scientists and agricultural scientists who want to share data securely with each other, and then do analysis on that data. And so it really has been instrumental in allowing for sharing of data between the private sector and then public sector scientists. And that's the primary use of it at this point, though we do see sharing within university settings as well. The sensing and actuation work, which I'll talk about in a little bit. Our application programming interfaces work is where we give kind of machine-to-machine communication and opened up for working with both internal partners and external partners. So you can imagine, instead of running a model on your individual computer, we run a model in the cloud and then allow people to access that over the internet. And then lastly, we do a lot of consulting, both in the nonprofit and in the private sector. So I'm going to shift gears now. I want to drill in on one part of kind of the GEMS ecosystem. And really our goal with GEMS sensing and actuation is, to the extent possible, automate the process of data collection throughout food and agriculture. And so that's kind of our big guiding mission. And it's a topic that I'm passionate about because, we hear from everyone from the Food and Ag Organization, United Nations, all the way down to talking, with scientists at the University of Minnesota, just the importance of data in driving, quote unquote, sustainable development broadly in agriculture. But the thing with data, and as all of you know, is it's really costly to collect. It's really time consuming to collect, and it can be one of the most error prone aspects of our work. And so what I'm going to do now is share a little bit about what we've been doing to address some of these challenges. So, I'm going to take you back to 2018 in the spring there. I started having in-depth conversations with Brad Morris, who's a principal AI strategist at BWCS Corp, a small startup company focused on AI and connectivity. So our expertise and interests were complementary. Our conversations focused broadly on the areas of ag tech. And then we started to really talk with agricultural scientists across the University of Minnesota to try to identify specific problems that they were having, and they were seeing. Data collection and transfer problems were ubiquitous. So in general, they had access to sensors, they had access to satellite imagery, they had access to drone imagery, but they were too expensive and difficult to use, and getting the data off of the devices onto somewhere where it could be analyzed was always challenging. And so the result was that scientists really weren't able to collect the data at the resolution that they needed. And so BWCS Corp came to visit the University of Minnesota. So this is Jim Wilgenbush, who's the director of research computing, was the head of Minnesota Supercomputing Institute, and is a co-director of GEMS. And then this is Brad Morris. And so they came, we met, we learned about one another. And after this meeting, we decided to sign a letter of intent to collaborate. And then we really quickly built a first prototype of this system to really kind of get after it, just a high level, do we think it's even possible to meet the objectives that are being laid out. So we built this 3-node system, so you can see how small these are. And then it live streamed data back over through this platform called the Things Network, and then into a database. And here's some pictures of it deployed. This was all deployed in California. and you know, just an idea of what the UI looked like. So it was very kind of crude in a lot of ways, just a first pass. And really what we wanted to learn some things. One, we learned about cost. Each of these nodes we could produce for under $30. The nodes, though, they had limited functionality. So we were just collecting air temp, humidity, barometric pressures. And then on one of the nodes, also the location of it. And then we learned that the connectivity of the system was quite good. And that we also, you know, kind of had some learnings around the different types of systems that we could build, which I won't go into right now. So in summary, at, you know, kind of at this point in the story, we moved from kind of just this bare bones idea of what we might be able to do, and we tested it really quickly to get what we would call a minimum viable product. And then from there, we worked with the leadership of GEMS to approach the Minnesota Ag Experiment Station for some seed funding. And what that allowed us to do was to do a 50 node sensor array at the Wasika Experiment Station last summer. And so to give you an idea, we put one of these nodes, so we did a substantial redesign of the nodes. This is all in collaboration with Pete Marchetto, his instrumentation lab. We manufactured all of these in-house, and that was a substantial undertaking. This is Bryan right here, who is a wonderful undergraduate research assistant in my lab. And you can just see, you know, you get this volume that happens. And here's a look at what it was like in the field. Our main focus here was to sense right at the soil surface and then below canopy, and that was really driven by collaborators Greg Johnson, Axel Garcia, and Forrest Azuno. This was deployed in the long-term agricultural research network plots. Just some more pictures so you can kind of see what things look like. So last summer, we learned a lot about how to make this hardware robust. We learned about the challenges of manufacturing nodes at scale. And so we've since gone and garnered additional funding. And we now have work across many different kind of application areas. So one of them is irrigation management, which I'll talk about a little bit here. We're also doing large-scale deployments of weather stations across Malawi and Africa. And then we're also doing a fair amount of work on envirotyping. And that's really with Eric Watkins' turf program here. And so we kind of have this growing portfolio of things. So I'm going to go a little bit into the irrigation management aspect of what we're doing. And it builds on what Vasudha was mentioned about in her talk. So what we've been doing is partnering with Vasudha to really test our system side by side with the other irrigation management systems that she's working with. And so these are deployed in Becker, Minnesota. There's 16 nodes. They're collecting soil moisture and temperature at 3 depths, and they're getting a reading every 15 minutes. What we're seeing at this point is that we have a big cost reduction in terms of the cost of the nodes being deployed in comparison to what's currently available on the market. And so we think there's a big opportunity there, not just only from a research perspective, but then also in potentially in partnership with farmers. To give you an idea what the system looks like, you know, so front and back on the inside. And then here's a look at what it was like. And these pictures are compliments of the Sioux. So you can see here, this specific sensor is an off-the-shelf sensor by a fun name of a company, Vegetronics, and this is the VH400. And you can go Google that if you'd like to. And you know, you have 3 of them then attached there. So just give you a sense of how big the thing is in row. Give you a sense of what they look like installed. So I think the big thing that's exciting for me and for Vasudha and others on the team going forward is that we've been recommended for funding from LCCMR. And what that will allow us to do is expand the irrigation management assistant tool. So we'll be improving the modeling that's going on in the back end associated with timing of irrigation and then yield prediction. But then we also get to expand out kind of to about 150 nodes. And they'll be split up on hopefully on farm and also on station. We will be, you know, assuming that this does go through and it's been recommended for funding to the legislature, but the legislature still has to approve it. approve it, we'll be looking for farmer partners who would be interested in potentially having nodes deployed on their farm. And so if this is something that you're interested in, please email me, because we would love, I assume that it will be funded, but you never know until things are finalized. But either way, it would be wonderful to start interacting, and we do have other projects that are spinning up as well on top of this. So with that, thank you so much for having me here today. I hope that this was a good high-level overview for you, and I'd be happy to take questions about any of the specific things during the Q&A time here.
If anybody has any questions for Bryan, please enter them in the Q&A. In the meantime, Carl, you can get your presentation loaded on the screen. Thank you, Bryan.
Thank you.
Okay, we're going to turn it over to Professor and Extension Nutrient Management Specialist Carl Rosen, who will be presenting Enhancing Soil Health in Irrigated Potato Cropping Systems.
Okay, you can see my screen now?
Yes.
Great, that's good. Thank you very much for the invitation to be at the virtual field day today. As Emily said, it would be much nicer to be out in the field giving this presentation, but nonetheless, I'm going to switch gears a little bit, talk about potatoes. It's an area that I've been working on for many years. And specifically, I'll be talking about soil health in relation to potato production. So some of the topics I'm going to be going over, we recently received a fairly large grant, a federal grant, to look at soil health and potato production. I'll go a little bit into the background of that, some of the objectives. We'll get into some of the challenges associated with potato production, particularly when we talk about soil health. Talk a little bit about measuring soil health, and then a little on-farm comparison, looking at an old potato field versus one that was never been in potatoes. So that's kind of the outline. So the proposal that we submitted was back in 2018. It was to the Specialty Crop Research Initiative. It's a joint proposal with 10 states involved. So we're really looking at a very large platform of data to try to help define soil health when it comes to potatoes. It's a 4-year project. And yeah, it spans a wide range of growing conditions, mostly irrigated. There are 24 collaborators, and I happen to be the lead PI on this. So I feel like I'm herding a bunch of cats most of the time, but we are getting a lot of work done. We started in September of 2018, so we're in our second growing season right now. There are 4 project objectives for this project. The first one is what we call our small plot objective, where we're looking at various management practices to optimize soil health. And this is one of the plots that we have at the Sandplain Research Farm. I'm not going to talk a lot about the details of that one yet because the data is still coming in. We also have some on-farm studies where we're looking at spatial variation of various soil health-based indicators associated with potato crop health, yield, and quality. And I'll give you a kind of a flavor of the type of data that we're collecting on those particular sites. And then the third and fourth objective. The third objective is basically the economics of soil health. So we're trying out a lot of different management practices. And the bottom line is it economical for the growers to use it? So we have an economics team working on that. And we also want to make sure that the information gets out. We have a website, and we're going to be doing some webinars for potato growers and things like that on soil health down the line. So that's an ongoing process as well. Okay, so the principles of soil health, I talked about soil health, but what do we really mean? And, you know, NRCS has a definition of it. You've probably all seen that, but some of the key principles are to keep the soil covered, you know, make sure that you don't have a lot of bare soil there, minimize your tillage as much as possible, increase crop diversity in there, kind of have a lot of rotation, keep living roots in the soil, so have cover crops whenever possible, keep that soil more alive, integrate livestock, which means that you're adding carbon to the soil. That's a very good way of doing that, although in some cropping systems it's more difficult. And then manage pests using ecological approaches, so minimize using pesticides and those types of things. So the challenge is for potato production is how can we adhere to some of these principles, and I'll get into some of those challenges in just a little bit. So when we look at potatoes, a major focus of soil health in general is to enhance the organic matter fraction. And this in turn is beneficial to the soil microbial community, particularly bacteria and fungi. That's what's the living portion of that soil. However, for many of you that know about growing potatoes, soil-borne diseases are a major concern. And so fumigation is used to control those soil-borne diseases. Some of the impacts of fumigation are beneficial in that you have a healthier root system, you often increase yield, but fumigation is non-specific. It kills not only the pathogens, but it also kills the beneficial organisms as well. Here's some data that we collected over the past few years, looking at the effects of fumigation and nitrogen management or nitrogen rate on potato yield. And in this, on the x-axis is the amount of nitrogen applied, on the y-axis is our tuber yield. And obviously, as you increase nitrogen rate, you're going to increase your yield up to a point. But the big dramatic effect of this graph is the gold line there is the non-fumigated plots, and you can see a significant yield drop at all nitrogen rates without fumigation relative to with fumigation. We also saw that you could actually get by with a little less nitrogen. Your optimal yield was a little bit lower with fumigation. So we think that's due to a healthier root system and the plant's able to access that nitrogen a little bit better. So you can see some benefits of fumigation. Unfortunately, it also affects the microbial community. So let's talk a little bit about measuring soil health, and I'll get back to this fumigation example in just a little bit. There's lots of indicators that have been proposed. Some are the chemical indicators, which some are just using our conventional soil tests, which is fine, our acid extractions to help predict what chemical properties of your soil. There's a newer test called the Haney test, which has dilute acid extractions. It's not well calibrated for Minnesota, and we honestly don't see a lot of difference. I mean, the numbers may be a little bit different, but the results, and in some cases, the Haney test doesn't perform as well as the conventional test, depending on the nutrient. So I would recommend continuing with the conventional test for chemical properties, but then we're looking at physical properties, the aggregate stability, which may not have as much applicability on sandy soils, but there are microaggregates, and we are in our study seeing some effect on some of the micro aggregates. So also how well the soil water infiltrates into that soil. We can look at penetrometer tests and textural class of that soil or of physical properties. And then finally, this is the biological properties, which are properties that are not as readily measured, at least in the past. They're becoming more popular now. Things like CO2 respiration, which gives you an idea of the microbial activity in that soil. looking at various carbon fractions in the soil, total, active, and water soluble, and then different nitrogen fractions is also being measured. And then finally, and one of the objectives of our particular project is to look at the microbial diversity, the microbial community, or the microbiome. And so here we're actually measuring the DNA in the soil and seeing how our management practices affect the DNA and in turn our potato production. So getting back to that fumigation effect, what we found, again, this is a graph showing various times after fumigation, and it also shows the microbial activity as measured by what was called the Solvita test. And as you can see, prior to fumigation, very little, I mean, basically it was the same as you would expect in terms of CO2 release. But with fumigation, you see a much more dramatic decrease in microbial activity relative to the non-fumigated soil. Even though, if you remember, our yields were better in our fumigated crops, our microbial activity was hindered. And so by the end of the season, you can see it's starting to recover again. But again, fumigation has very dramatic effects on the microbial populations. And so what we're looking at is trying to see if there's some management practices in addition to fumigation that might help enhance these the microbial fraction of that soil. Okay, so getting to the real challenges in potato production. Reduced tillage or minimizing tillage during the potato year is very difficult, just because of the way you plant potatoes and the way they are harvested. It's virtually impossible to do that. Integration with livestock is also difficult. There's usually not a lot of animal operations around, but you know, it is becoming an area where some of the inputs may be that adding some of that carbon might be useful. And then again, as I mentioned, fumigation and low residue, crop residue after harvest is also a challenge. So what are some of the solutions that can be used? Use cover crops after harvest of the rotation crops. So things like rye and oats are very beneficial. Disease suppression, if you can use mustards, and for compaction, oilseed radish tends to help. So there's some cover crop options that we're looking at. We're looking at some composted manure additions, reducing tillage during the rotation year, since it's not as easy to do during the potato year. And then with the rotation, try to use some high rotation crops, things like corn and small grains are good. And then we're also looking at the possibility of various inoculants after fumigation. So those are some of the things that need to be looked at. Okay, so in getting back to our project, we're basically looking to see if there's specific management practices that can promote beneficial microbial populations, and we want to know if they're associated with suppression of disease and increased potato yields. So those are some of the objectives of this particular project. So for objective one in our small plots, I'm not going to show any data, but just to give you an idea of the type of information that we're going to generate. We have 2- and 3-year rotations. It's a 4-year project. So we're in our, we started a 2-year rotation this year, and then the final rotation, or the final potato year, will be in 2 years. So we're looking at effects of fumigation. We're looking at some carbon additions like composted manure, various rotations. We have our conventional corn soybean rotation. We're looking at a corn pea mustard rotation. We're looking at cover crops like rye versus mustard. We also have cultivar, potato cultivar in there, and we're looking at some inoculants as well. And what we're going to be measuring are things like potato scab and verticillium. The soil-borne disease that we're most concerned about is verticillium. So it has dramatic effects on yield. So the second objective that I'm just going to move on very quickly is this spatial distribution, or the spatial effects on-farm soil health-based indicators associated with potato yield and health. And what we are doing in that, we selected 4 fields in Minnesota, and I'm just going to focus on the first 2, which is a new field, which is not fumigated and hasn't been in potatoes, at least that we know of. Then we're comparing that to a field very close in proximity next to it, which is an old field that was fumigated. Okay, so this gives you an idea of what the layout looked like. So the new field was cleared in 2017, planted to soybeans in 2018, and then planted to potatoes in 2019. And these are the grid points. Each point we took a sample in both of these fields, and they're both about 14 acres. The old field right here has been in potatoes for over 40 years in a 2-year potato rotation. So a lot of history of potatoes there, and has to be fumigated to get good production. So our yields are right here. Oh geez, I forgot to put the average yield. The average yield in the new potato field was lower than in the average yield in the old potato field. It was about 355, 365 for the new field, and 385 for the old field. And this was surprising to us because we thought there would be no diseases in the new field, and the old field would have had more diseases, even though that it was fumigated. So one of the parameters that we measured was soil organic matter. And you can see that the soil organic matter, which is often associated with good soil health, higher organic matter, usually is better. And we found that in the new field that hadn't been cropped for many years, had about twice the organic matter that the old field had. So that is not too surprising, given the fact that you're tilling the soil many years, over 40 years, that organic matter will drop. Okay, so one of the surprising results that we found is that when we correlated total yield versus organic matter, the actual yields tended to go down with higher organic matter. So that was a little surprising to us, both in the new field and maybe to a little lesser extent in the old field. So why was that? What we did was we looked at a digital elevation map, and what we found is that there's more depressions in the new field than in the old field. And what happens when you have depressions, there's about a 10-foot difference between those depressions and the higher ground. And what happens, you get accumulation of organic matter in those depressions, but also associated with those depressions are poor infiltration of water. You just don't have the best conditions for root growth when you have those depressional areas. And that's kind of what we saw here. Again, there's a lot of variation, but in general, as you increase in elevation, your organic matter tended to go down. You had more organic matter in your depressional areas, and that's where we had our lower yields. So the fact that we had, this negative effect with organic matter, which is again supposedly a good indicator of soil health, in this case you have to put it into context. And so I think that's the main point that I want to bring out in this presentation. So kind of the summary and key points is that soil health is the result of a complex set of interactions among physical, chemical, and biological factors. For potatoes, you need to concentrate in the rotation years, look at cover crops, reduce tillage, high residue crops, add manure or compost if available, and test inoculants after fumigation for viability. And it's a long-term commitment. You're not going to change soil health overnight. There are no silver bullets. It's unlikely that any one soil measurement will be able to define a healthy soil, as we saw with that organic matter example. So we do hope the results from this project can be used to help identify kind of a suite of metrics and to help guide our management practices and improve potato yield. So with that, I think I'm finished. May have been a little bit longer than I expected, but if there are any questions, I'd be happy to answer them.
Thank you, Carl. It doesn't look like we have any questions coming in right now, but if anybody has some later, we can come back to that. We're gonna move on to Associate Professor and Extension Soil Fertility Specialist, Fabian Fernandez, will be presenting lessons from current nitrogen management research projects on irrigated corn.
Thank you, Emily. Let's see, I'm trying to get the screen here. Okay, let's see. Do you see my full screen here?
Yes.
All right, wonderful. All right, thank you very much, everyone, for attending today. My name is Fabian Fernandez. I'm a nutrient management specialist and in extension and research. And today I will be talking a little bit about some of the current ongoing projects, as well as some recently finished projects that I am doing in irrigated sands, mostly irrigated or non-irrigated sands. And we'll start, let's see. All right. Do you see the full screen? Because I'm trying to put it in presentation mode here. There we go. All right, so this first study is a long-term nitrogen study. This is funded by the Fertilizer Research and Education Council, AFREC. And it's located in several different parts of the state. We have a corn-soybean rotation. And the rates are listed there. We have 5 rates of nitrogen. For the location at Becker, this is the one study that I have at the new farm in Becker. We have those rates apply for corn or corn soybean, and we apply them at in 3 splits, basically, with the same amount on each split to complete the total at V2, V5, and V8 development stage. And then at the other locations throughout the state, we have locations in Crookston, Morris, Lamberton, Waseca, and Rochester. We apply the nitrogen rates, the 5 nitrogen rates, all at pre-plant. The rates that you see here are for the sandy soils, which are higher rates than we have for the other locations that are non-sandy, non-irrigated sites. And the idea here in this project is to, well, first of all, establish a really strong baseline of soil sampling so that we can look at the physical, chemical, biological properties of the soil. And then the idea is to establish these plots. We started this in 2019 and continue them for at least 10 to 15 years. And applying these same rates year after year. And then we would like to look at the changes that these nitrogen fertilization rates had on the soil, so the physical, the chemical, and the biological properties, and compare those to the starting conditions. The other part is that all of this data allows us to create response curves, and we can use those response curves to update the MRTN, the maximum return to nitrogen rate calculator, that we have to guide applications across Minnesota. And then the last objective of this project is to, after 10 to 15 years, is to impose new nitrogen rates on top of those treatments. So basically we establish this. We will hopefully over time we'll see changes that happen because of this continuous fertilization, and then we will be able to look at responses when we change the rates on those previous conditions, and seeing how that impacts nitrogen management. So it's really a long-term study, and I am very thankful to AFREC that had the vision to support this project. Long-term projects are, you know, they're really an investment, and they're willing to do that. And I'm very grateful for that. There is a large team of researchers involved in this project as well. And just to show, these are the results from last year. We have, for each of those 5 N rates, and I should have mentioned these in the previous slide, here in the bottom you can see what one replication looks like. So for each of the rates we have 6 plots, and these are replicated. Each one of these schemes of 5 rates are replicated 4 times. So you end up with 5 rates with 6 repeats replicated 4 times. And so that allows us to look at the response curve. Here is for 2019 in continuous corn on the top. You can see the response that we got. The EONR is the green dot, and for continuous corn, the EONR, or economic optimum N rate, at that 0.1 nitrogen to corn price ratio was 240 pounds of nitrogen per acre, and the yield at that EONR was 219 bushels. When we look at the guidelines from the MRTN calculator, the maximum return to nitrogen calculator, we recommend 209 pounds of nitrogen with a range of 192 to 225 pounds. We see that the EONR here was about 31 bushels more, or 31 pounds more nitrogen than the MRTN calculator. And I think this reflects the fact that we had a really wet growing season last year. And even though we were applying nitrogen, we did lose some of that nitrogen. When we look at the lower graph, this is for corn after soybean, we again see the same thing, that we got the EONR at 200 pounds, which is about 21 pounds more than what we would recommend with the MRTN calculator. But the important thing of this is that it allows us to keep adding some of this data into the database, and it allows us to have more and more different treatments or different growing season conditions into this calculator, so that it gives us a more robust data set. As I mentioned, we have 6 sets of plots there with each of the rates replicated 4 times. And so those response curves that you saw there were average across all these 6 sets. But what you can do too here is create those response curves 6 times for each one of these sets. And so this allows us to look at the variability in economic optimum N rates, and that's what you're seeing here for continuous corn in the brownish colors, and the reddish is for corn after soybean. The first row, or the first row is EONR, and the second is the yield at the EONR for each one of those 6 sets. And you can look at the mean, the median, and the range, and you see here immediately that there is quite a bit of variability in the EONRs. And this represents basically for each one of these rotations slightly more than 1.5 acres. And so you can see that within just 1.5 acre, the range for continuous corn in the EONR was 55 pounds of nitrogen. For corn soybean was 68. And so again, it shows the importance of using the MRTN, but also the importance of realizing that there is variability, sometimes a very small scale variability in terms of the EONR. So those are kind of the first results that we have from this project. This is another project that was concluded a couple of years ago. This was funded through Pioneer, Cortiva now. And by the way, I put the funders, or the funding sources, in the slides to acknowledge the funding for each of all the projects that I'll be presenting today here. This project, we were interested in looking at the effect of pre-plant only application compared to a split application. And so Again, we had a number of nitrogen rates depicted by the different dots in the graph. The pre-plant application is the blue line, and the yellow dot represents the economic optimum end rate for that application. The orange dots represent the application where we apply 40 pounds pre-plant, and then we apply the same amount of nitrogen to match the pre-plant rates at around V8, V9. And this was all done at the Becker farm, the old site. And you see here that the split application by far was the better application. It was way better than a pre-plant only application. It yielded more. It required just a little bit more nitrogen, but it yielded substantially more. And so based on these and many other projects that we have done over the years, we definitely recommend that in irrigated sands that the nitrogen should be split applied. We also had, with the black dots, a few rates where we apply 80 pre-plant and then side dress at those rates. And again, we saw little difference compared to only applying 40 pre-plant. So which shows that the plants don't really need a lot of nitrogen to get going, but with a small amount of nitrogen, they will be okay, but the split application is the important thing. This is another project that was also conducted in Becker. It's a study that is finished already. This was funded by the corn growers. And what we are looking at in here is the first several treatments that are on the left are different nitrogen sources. They were all applied pre-plant, anhydrous ammonia, anhydrous ammonia plus N-serve. And then 3 different ratios of ESN and urea, 40-80 or 80-40 or 100% ESN or 100% urea. And these are the yields average over 3 growing seasons and 4 site years of data. What is very clear in here is that the urea pre-plant application was the worst kind of treatment that you could have in there. And you could see that when we look at the different ESN to urea ratios, as we increase the amount of ESN, we increase the yield for each one of those. But regardless of the source of nitrogen that we use pre-plant, you can see that when we did a split application where we apply 40 pounds pre-plant and then 80 pounds side dress, and these were all at 120 pounds. This was a very small rate of nitrogen, suboptimal for sure. The idea was not to maximize the yield, but it was to look at the differences due to treatment. We see that with those split applications, we increase the yield substantially, much more so than any kind of source of nitrogen applied pre-plant. This is moving on to, as I mentioned, this is going to be a little bit of a marathon because I have several different projects that I am presenting. This is a study in Staples, Minnesota. This was funded by the Minnesota Department of Agriculture, where we were looking at EONRs and yields at the EONRs, and then looking also at some of the residual nitrogen in the soil when we have either irrigated or non-irrigated conditions. And in general, you can see this was done over a 4 growing season period, 2015 through 2018. We find that in general, the irrigation substantially increased corn yields, the end use efficiency, and the economic optimum end rate stability. When you look at the range of values for the EONR in irrigated, you had about a range of 62 pounds of nitrogen compared to 127 pounds for the non-irrigated. So this study really shows that irrigation makes a huge difference, not only in the yield, but also in the use efficiency of the nitrogen fertilizers that we apply. So definitely a great advantage to have irrigation in these sandy soils. And then this is the last study I would like to touch on. This is a long-term study. We started this in 2011. It's a partnership between the Pope County Soil Water Conservation District, the Minnesota Department of Agriculture, and the University of Minnesota. And what we have in here is a series of nitrogen rates with cover crops. We have 5 nitrogen rates with winter rye cover crop, no rye cover crop, and then we have 2 rates of nitrogen, a check and around the economic optimum end rate with Cura clover, which is a living mulch. What you see here is an aerial image of the site. We have continuous corn on the right, and then a corn soybean rotation on the other 2 sets of plots. You also notice that there are little pipes sticking out of the ground. Those are kind of like what Vasu was talking about with the permanently installed lysimeters. We have lysimeters in many of these plots, so it allows us to look at the water quality. And we strip till this site so that we can have a living mulch, so plants that stay alive year round. And so we are looking at the effects of nitrogen rates and these cover crops on both improving water quality, the environmental aspect, and also in terms of agronomics, how much nitrogen we would need if we have these living mulches or these cover crops in the system. So jumping in quickly on some of the results, here we are looking at the season-long concentrations, nitrate concentrations in the water that passes the root zone and eventually would end up in groundwater, or some of it will end up in the groundwater at least. We have, this is the average across 2016 through 2019. In the top table, you see the N rates for the 3 rotations, continuous corn, or cropping systems, I would say continuous corn, corn, soybean, and then the soybean phase of that rotation. You see that in general, the concentrations, nitrate concentrations increase with the increasing nitrogen rate, but you also notice that in the soybean crop portion of the rotation, the concentrations are almost as high as the ones where we actually apply nitrogen, and we are just basically we are not applying any nitrogen to the soybean crop, but we still see the concentrations are substantially higher, relatively similar to where we apply nitrogen for the corn crop. And then on the bottom table, we see the effect of the cover crops. We see that in general, there was not a huge amount of difference, especially when we look at rye, no rye, there were basically no differences, but the Cura clover substantially reduced the concentrations by 52% in continuous corn, 73% in corn soybean, and 72% in the soybean corn rotation. So a substantial reduction compared to the rye or no rye treatment. So a great advantage there. Then we look at the same data now, but looking at the load. So these are pounds of nitrate nitrogen per acre that are past the root zone. And we see that when we look at the economic optimum end rate, so 225 pounds for continuous corn and about 180 pounds for the corn soybean rotation, when we go above that, we don't really see a substantial increase in the loading that we have when we apply more nitrogen. And we also see that in the soybean crop where we apply no nitrogen, the loads are almost as high as some of the loads that we see in the corn system. And then when we look at the effect of cover crops, so again, bottom table, rye, no rye, and the Cura, no difference between the rye and no rye in terms of reducing loading, which is unfortunate. But we see the Cura clover had a substantial benefit. In fact, when we look at the Cura versus some of the rye treatments or no rye treatments with no nitrogen apply, the Cura actually reduced loading more than the check plots that receive no nitrogen. When we do an average of the rye, no rye, compared to the Cura clover reduced the load by 66% in continuous corn, 79% in the corn soybean rotation, and 77% in the soybean phase of the corn soybean rotation. So a substantial reduction in loading. And then if we move ahead and now look at the more of the agronomic aspects, we see that with nitrogen rate, we increase yield. So there was definitely a response to nitrogen in the corn system, the continuous corn cropping system, and the corn soybean. There were no differences in the soybean yield, looking at the residual effect of those nitrogen rates. But then, and I'm not presenting this data, but when you look at the EONRs, or the economic optimum end rate, to these end rates, we find that even though there was no difference when we look at rye versus no rye in the yield, at the economic optimum end rate, we find that in continuous corn, we needed more nitrogen. I think it's 24 pounds more nitrogen was needed for the economic optimum end rate at continuous corn. And in the corn soybean, there was really no difference between rye and no rye as an average across all the years. But when you look at all the years, in all the years except one of the years, you actually on average needed about 12 pounds more nitrogen when you had rye in the system in the corn soybean rotation. And then when we look at the yields for the cover crops, as I mentioned, rye no rye made no difference, but Cura clover unfortunately reduced the yields substantially compared to the average of the rye no rye treatments. 36% reduction in continuous corn, 38% reduction in corn soybean, and even in the soybean crop where we are not doing anything with nitrogen, a 19% reduction in the yield. So even though Cura clover is great at reducing loss or nitrate leaching, it also competes pretty heavily with the grain crops. And so as a summary, using emojis as a way to kind of restate the results for this study, winter rye cover crop had produced basically no benefits in reducing nitrate leaching, which is very unfortunate. But the good, I guess, somewhat good thing is that it did not really impact grain yield, but it did increase the EONR. So we need to apply more nitrogen when we have rye in the system. One other thing that we had a challenge with in most of the years is in the establishment phase. We try to plant this winter rye cover crop in the fall, and sometimes we have good establishment. Some years we don't have as good of establishment. And then in terms of the Cura clover, we observed that there's a substantial reduction in nitrate leaching, which is wonderful, but we also observed that it competes very heavily with the crop. And so we need to continue to refine, and that's some of the things that we are doing right now, keeping our fingers crossed that we'll be able to make the system work for us so that we retain the benefits of nitrate leaching reduction, but maybe have less of a hit in the yield. So with that, I'll stop, and if there is time, Emily, I will answer any questions.
Matt is wondering, for the 15-year study started in 2019, how many years of data are needed before extension fertilizer recommendations can be revised or updated for farmers? 2, 5, 15?
Yeah, that's an excellent question, and it's what we are doing is we are always adding this data into our calculator. For the non-irrigated sites, actually, we are doing this every year, and over time, you build a fairly large database. So 1 year of data doesn't really impact the change, or it doesn't change things very much. In the sandy soils, irrigated sandy soils, we are looking at increasing the number of sites, and this certainly will help us with this study and a few other studies that we have in order to kind of generate enough data that we can kind of add it every year. I would say that, typically when you look at enough data, I would say 3 to 5 years of data, when you have enough response curves is where you start to get enough information to have a robust data set to look at these things. So we continue to monitor these things every year. And again, you need to look at across years, because as I mentioned, 2019 was a really wet year. And so I was not surprised that the end rates were a little higher than the recommendations. As you remember, probably we updated the recommendations in 2015. It had been a long time before that since we had done any changes. And part of that was that we didn't have a lot of data. Now we are generating slowly a little bit more data in the irrigated sands. And so I'm hoping that we'll continue to look at these things kind of in a continuous basis.
Okay, and then one more question from Paul. Why is the Cura reducing yield?
So I think the Cura is reducing the yield because it does compete for nitrogen in the crop. One of the things that we are doing now is, for the first few years, we were really trying to establish the Cura clover system. And so we were trying to kind of favor the Cura clover. We were planting corn a little bit later. And we were trying to suppress the Cura before planting the crops, but we were doing some mowing, some low doses of glyphosate to kind of knock it down a little bit. But now that the Cura clover is very well established, we are actually, this year, we started applying like full doses of glyphosate right in the row and trying to really suppress that Cura clover so it doesn't compete as much. But the main thing is that these first few years were kind of establishment phase of the system. And the Cura clover, even though it fixes nitrogen, it does require nitrogen. And I think, or I'm hoping at least, that as the system becomes a long-term system, the Cura clover actually will become a contributor. So it will be fixing nitrogen for our crop rather than taking it away from the crop. But it's one of those things that we have to do the research to see how that works.
Okay, and then we'll do one more quick question before we turn it over to Jeff. Is there a cover crop that you can recommend for scavenging and following row crops?
Well, the best ones I would say are typically cereal crops like rye. There are a number of crops. There are people that are looking at different mixtures with deep-rooted cover crops and things like that. And the biggest challenge, and there are a few people in the university doing research with all these cover, and so, but really and truly is the establishment, because once you get the crop established and it grows, it will take up nitrogen. And so the challenge is that establishment. So I don't have a direct answer, but I would say any of them really work as long as you can establish them well in the fall, and then they survive into the spring.
Okay, thank you. We will get our last presenter up and running here. Jeff Coulter, Extension Agronomist and Professor, will be talking about advancing intensive management of continuous corn on irrigated sands.
Okay, well, thank you. It's a pleasure to be here today and present some of our research. So to give a little overview, one of the greatest things that we're faced with today in agriculture is meeting the new demands that society has for us. And those demands are that we have cropping systems that are both productive and environmentally responsible. Sustainable intensification is an approach to improving the yield and profitability of cropping systems with sustainable practices. As we know, nitrogen is typically the most limiting nutrient for corn. Application in excess of corn requirements reduces risk of yield loss, but with economic and environmental consequences. A few years ago, a small group of us got together and became interested in doing some high-yield continuous corn research on irrigated sands. Some of the questions that we wanted to address were, what yields are possible for continuous corn? How far are current yields from these levels? Is standard fertilizer management that is in line with university guidelines capable of attaining and maintaining yields at levels close to yield potential? And can advanced fertilizer management and sustainable intensification improve yield, economic return, and nitrogen stewardship? So in 2014, we started a study that was funded by the Minnesota Agricultural Fertilizer Research and Education Council. And this study went on from 2014 to 2019, and I'm going to present results from the first 4 years. This research was conducted at Becker on the University of Minnesota Sandplain Research Farm in collaboration with Jeff Vetch and Ron Faber at the U of M, and with Scott Murrell, who was with the International Plant Nutrition Institute. This was conducted on irrigated sands, and we had an initial, or a yield goal of 250 bushels per acre. We chose to do this research in continuous corn because continuous corn requires top management for high yields. Continuous corn is also at greater risk of nutrient losses since we are applying nitrogen every year. We also wanted to conduct this research on irrigated sands because irrigated sands have high yield potential, but at the same time, they have greater risk of crop nutrient deficiency and greater risk of nutrient losses. In this study, we looked at a couple different factors in our treatments. The first treatment factor was agronomics. We looked at normal agronomics versus intensive agronomics, which we call sustainable intensification. And then within each of these two agronomic practices, we looked at either standard fertilizer management or advanced fertilizer management. And then within each of these, we looked at plots that had nitrogen applied versus no nitrogen applied with the same of all of the other factors that were applied in those treatments. So we ended up having 4 treatment combinations, and then within each of those, we looked at with nitrogen and without nitrogen so that we could calculate indices of nitrogen use efficiency. Now, the components of these treatments, the agronomics and the fertilizer strategy, were developed in collaboration with crop advisors, industry agronomists, and farmers. The soil characteristics at our site were a Hubbard-Mosford loamy sand. The treatments were applied to the same plots each year, and the initial 0 to 6 inch soil test levels were a pH of 5.6, which was a little low, so we applied some lime before we got started with the experiment. The Bray 1 phosphorus was 33 parts per million, which is very high according to the U of M guidelines. Potassium was 59 parts per million, which is considered low, and zinc was 2.0 part per million, which is considered sufficient according to the U of M guidelines. This slide breaks down the different factors of the normal agronomic versus intensive agronomic treatments. In this system, we used a disc script tillage system for all of the plots, and the agronomic treatments, normal versus intensive, differed in corn residue removal, hybrid maturity, and planting rate. In the normal agronomic system, we did not remove corn stover after grain harvest in the fall. In the intensive agronomic system, we went in and removed 40% of the above ground residue after grain harvest and also before fall tillage. The agronomic treatments also differed in hybrid maturity and planting rate. In the normal agronomic system, we chose a mid-maturity hybrid, a 97 relative maturity, and we planted at 36,000 seeds per acre. In the intensive agronomic system, we planted a 103 relative maturity hybrid at 41,000 seeds per acre. This slide breaks down the different fertilizer management treatments. We had standard fertilizer management versus advanced fertilizer management. And this shows all of the different levels of fertilizer that were applied. The main thing is that the two treatments differed in phosphorus application and also in the time and form of nitrogen application. So in the standard fertilizer management practice, phosphorus was applied according to U of M guidelines, and according to those guidelines, no phosphorus was needed since we were already at a very high level. In the advanced system, we applied phosphorus at estimated grain removal rate. So we calculated that based on a 250 bushel per acre yield goal, and that was 90 pounds of phosphorus, P2O5 per acre applied annually. In both treatments, we applied 100 pounds of K2O per acre annually, and we also applied 20 pounds of sulfate sulfur per acre each year. We also applied 4 gallons of 1034O starter fertilizer in the furrow at planting for both treatments. Now we can look at the different forms and rates of nitrogen that were applied. In both treatments, we applied 210 pounds of N per acre, but the timing and form differed a little bit. So in the standard fertilizer management practice, we applied 40 pounds of N per acre side dressed as urea at the V2 stage, and then we applied 165 pounds at the V6 stage. In addition, we had a little bit of N applied in the furrow at planting. In the advanced fertilizer management treatment, not only did we have the 1034O in the furrow at planting, we also applied surface banded starter placed 2 inches to the side of the row on the soil surface at planting. We applied 7 gallons of 2800 and 2 gallons of ammonium thiosulfate, 120026. We did not apply anything at V2. We applied 70 pounds at V6, 76 pounds at V12, and 35 pounds of N per acre at tasseling. And these timings and rates were based on what we expected to be closer to what we could expect corn to be taking up. So we tried to time our rates and timings so that they would better match corn and uptake based on what we know about how a corn plant grows and develops. This slide shows the yield results. So what we have in this first row, it shows the different agronomic treatments. So we had normal agronomics versus intensive agronomics. And remember that the intensive agronomics had partial stover removal, it had a higher planting rate, and a longer maturity hybrid. Then within each of those, we had standard versus advanced fertilizer management. In this figure here, we're showing the yield levels for each of the 4 years and also for the 4-year average. Within a row, yield levels followed by the same letter are not significantly different. And I've highlighted those cells that are the treatments that have yields that were statistically the highest among the treatments that were evaluated. One thing that we can do with this data is we can compare the highest yielding treatment versus the lowest yielding treatment. And if we do that, we see that we had a yield gap of 40 to 59 bushels per acre. Consistently, the lowest yielding treatment was the treatment that had normal agronomics and standard fertilizer management. And the treatment that had the highest yields was intensive agronomics with advanced fertilizer management. We also saw that we had moderate yield with either intensive agronomics or with advanced fertilizer management alone. So if we compare the base treatment, the normal agronomics with standard fertilizer management, and we look at increasing just the fertilizer management, that gave us a yield boost. And similarly, if we compare the base treatment to just increasing the agronomics, keeping the fertilizer management the same, we also see a yield boost. And those treatments provided somewhat moderate yield. We also conducted a detailed economic analysis. So for this economic analysis, it's called the partial budget analysis, which means that we just took into account those costs and prices that differed among the treatments. And here it shows the costs and prices that we used for the different levels of agronomic management and the different levels of fertilizer management. Remember that the intensive agronomics had partial stover removal, so we had to account for that cost. It also had a higher planting rate, 5,000 more seeds per acre. So we accounted for that. So intensive agronomics has a little higher seed cost. We kept the seed cost for the 2 different hybrids the same. And then we can look at some of the other costs that differed among treatments. Particularly, all of the costs below this dark line here are for the differences in the fertilizer management. And what we see is that the advanced treatment had almost $51 more per acre for phosphorus fertilizer. It also had some cost for the band applied fertilizer applied at planting. No cost for a V2 application because V2N was only applied in the standard treatment. And it had differed also a little bit in some of the other costs based on when the N was applied. But remember that the total N rate between advanced and standard fertilizer management was similar. We can look at the total cost between the different treatments. So let's look at normal agronomics versus with standard fertilizer management. When we compare that to normal agronomics with advanced fertilizer management, the cost goes up by about $52 per acre. So this is just due to increasing the fertilizer management. We can also look at the base treatment and compare that with intensive agronomics, keeping the fertilizer management the same. And if we do that, we see that the cost per acre is going up by about $45 per acre. And when we add the two of those together, we see that we have a much higher cost for the intensive agronomics with advanced fertilizer management, around $91 per acre or so more compared to the base treatment. In this study, we also took into account the cost of drying the grain. We assumed 4.5 cents per point per bushel. We assumed $3.50 per bushel for corn across the board, and we assumed that the value of the corn stover that we sold in the intensive agronomic treatment would be about $35 per round bale. So taking this into account, we can calculate the partial net return. And this is based only on those costs and treatments that differ, costs and prices that differ among the treatments. We see we've got the two different levels of agronomic management at the top, normal versus intensive agronomics. Within each of those, we've got standard versus advanced fertilizer management. And then again, we've got the partial net return values by year. And the way to look at this is to look at it within a row, and values that are followed by the same letter are not significantly different. And we also presented the 4-year average at the bottom. What we see is that greatest partial net return occurred with intensive agronomics plus advanced fertilizer management in all 4 years and on average. However, intensive agronomics with standard fertilizer management also produced equivalent partial net return in 3 of 4 years. And if we just look at the average value over the 4 years, we see that partial net return was about $148 per acre greater with intensive agronomics plus advanced fertilizer management compared to normal agronomics plus standard fertilizer management. Another thing that we need to be looking at is indices of nitrogen use efficiency. We measured several of these, and I'm going to report the 2 of them that are most pertinent to growers and ag advisors, particularly agronomic nitrogen use efficiency and corn end recovery efficiency. So in this slide, we've got the different levels of agronomic management, normal versus intensive on the left side. Then we have either standard or advanced fertilizer management for both of those levels. And for this table, we want to look at it within a column. So we want to look up and down within a column, and those values that are followed by the same letter are not significantly different. So before I start explaining this table, I should first explain what these indices are. So the way that we calculate these indices are we look at a given treatment, for example, normal agronomics with standard fertilizer management. And then we look at the results from plots that had nitrogen applied versus the results from plots that had no nitrogen applied. And we can calculate the difference between those and use a calculation and come up with these indices. And we do that for both of these columns. And one of the indices that we look at is agronomic nitrogen use efficiency. And this is the increase in grain yield per pound of N per acre that's applied. And we see that average across the 4 years, the highest yielding treatment, the one that had intensive agronomics with advanced fertilizer management, had the greatest agronomic nitrogen use efficiency. And that's intuitive because that treatment had the highest yield, but it had the same nitrogen rate that was applied in these other treatments. Similarly, if we look at the 2 treatments in the middle, they had an intermediate agronomic NUE because they had intermediate grain yield, but had the same nitrogen rate that was applied as the other treatments. Another indice of nitrogen use efficiency that we can look at is called corn end recovery efficiency. This is a very important indice, and it's an indication of how much of that N was taken up in the corn plant at maturity, just prior to harvest. So this is all of the nitrogen that's in the above ground tissue of the plant, in the grain, in the cob, and the stover. And this indice is looking at the increase in that N uptake in the above ground plant material, the pounds of that compared to the pounds of N fertilizer that were applied. And what we see for corn and recovery efficiency is that we see a boost in corn and recovery efficiency when we go from standard fertilizer management to advanced fertilizer management. So we see that for both the normal agronomics, and we see a similar incremental increase with intensive agronomics. So those are kind of the overall results from the study. And our conclusion then is that intensive management systems with carefully chosen practices and rates of inputs can increase corn yield, profitability, and indices of nitrogen use efficiency. Thank you very much, and I would be happy to take questions if we have time.
Thanks, Jeff. You have one question that applies back to your fertilizer management slide, which I believe was number 10.
Okay.
The question is, the N amounts did not add up to 210 pounds per acre for both. Can you clarify, please?
Sure. So we have some N that is applied in the 1034.0 in the furrow. And if we add that up, that adds up to the 210. So 40 plus 165 is 205, and that would leave 5 pounds of N per acre in the 1034.0. Similarly, for the advanced one, if we add the 5 pounds of N per acre from the 1034.0, which I didn't show, mainly due to limitations in space, and if we also add the amount of N that was applied in the surface banded starter, I think that was around 21 pounds of N per acre or something. That would get us up to the 210. Sorry for that confusion.
Okay, if anybody has any other questions for Jeff, or I believe all the presenters are still on, and Ron from the Sampling Research Farm is also on. If you have any questions for anyone, please feel free to enter them now. In the meantime, I did put a evaluation form in the chat if you wouldn't mind going in and filling that out. Oh, we have another question for you, Jeff. Can you discuss how stover removal influenced your results?
Yes, I think stover removal had somewhat of an effect on the yield. So we noticed that we got a yield boost when we went from normal to intensive agronomics. And I think that yield boost when we went to intensive agronomics was due to the three things that were involved: the longer maturity hybrid, the higher planting rate, and the partial stover removal. And I think it was in that order that those factors influenced the yield level. I think the longer maturity hybrid had the greatest effect, followed by the higher plant population, and then thirdly followed by the partial stover removal. We chose 40% stover removal because some published literature has shown that in continuous corn systems, if we take off about 40%, up to 40% of the corn stover, that can, that's kind of the upper limit for the amount that we can remove without depleting soil organic carbon, while also leaving enough residue to protect against soil erosion. And when we start to take off some of that above ground corn stover, then the system kind of tends to act a little bit more like a corn soybean rotation than continuous corn. I would expect that the yield boost from the partial stover removal was probably up to 5%, but not likely more than that. And that's based on some other studies that I've done and other people have done that have looked at yield increases when we remove different amounts of corn stover in continuous corn.
Okay, if anybody else has additional questions, please enter them. If not, we will wrap things up. Thank you all for participating. We did record this session, so a recording will be probably sent out to everyone who registered, as well as put on the SPRF website. Like I said, there's an evaluation form in the chat right now. We'll also send that out in an email. Otherwise, thank you, everyone.